Hello all. Hello my dear students. Myself Archana and I welcome you all to this beautiful platform of Physics Wallah. As we know that term 1 are around the corner, so today we'll be revising the chapter Molecular Inheritance. No need to worry, just be focused throughout the lecture, go step by step with me, learn step by step with me and you'll be prepared for the term 1. So without delaying, let's start with the chapter Molecular Inheritance. So what are the goals of this chapter today? We will be dealing with the search for genetic material. We'll be dealing with the DNA, RNA and their structure. We'll be dealing with central dogma, packaging of DNA, replication, transcription, translation, genetic code, regulation of gene expression, lac operon, human genome project and DNA fingerprinting. So first of all, let's start with the search for genetic material. As the name of the chapter suggests, molecular inheritance. So, what is inheritance? As we all know, it is the passage or the transmission of characters from one generation to the other. But the actual question which arises in this chapter is that what is that biochemical which carries this information? So, let's find out the answer of this question. For that sort, let's go back to the Mendel's time. That is around 1860s when the laws of inheritance came. Mendel did not have any idea about gene, chromosomes, DNA. He used the term factors. And then after some time in the year 1900, the Mendel's work was rediscovered. And after the rediscovery of Mendel's work, everybody suddenly started to think that now laws of inheritance are in the picture, but who is that biochemical which carries this genetic information? So, the search for genetic material started in the year 1926. And the first prominent work in this direction came in the year 1928 with Griffiths' work. So, let's discuss Griffiths Experiment. So, Griffiths Experiment came in the year 1928, and for his experiment, he took the bacteria Streptococcus pneumoniae, or you can also call it as pneumococcus. This bacteria has two types of strains, that is R strain and S strain. Now, as we all know, basic structure of bacteria, that it has cell membrane, around it it has cell wall, and around it it has additional layer called as capsule or slime layer. But it is not necessary that all the bacteria should have capsule. So, R strain does not have capsule around the cell wall and S strain has the capsule. So, S strain forms the smooth colony and R strain forms the rough colony. S strain is the virulent strain. That means it is the pathogenic strain which can cause the disease. But R strain is the non-virulent or the non-pathogenic strain which cannot cause the disease. So now let's understand the experiment. For his experiment, Griffith took mice. And first of all, he injected the living R strain into the mice. And it is understood that the R2 strain cannot cause the disease, so mice was living. In the second set of experiment, he injected S strain into the mice, and obviously the mice would be dead because the S strain causes the disease. But in the next set of experiment, he injected heat-killed S strain into the mice. Now, heat-killed means inactivating the bacteria. And he inactivated the bacteria and injected into the mice, so mice was living. And in the fourth set of experiment, he took a mixture of living R strain and heat-killed S3 strain. So, what was anticipated? That R strain cannot cause the disease and S strain is inactivated. So, the mice should have lived. But to his surprise, the mice was dead. Why? Why it happened? R strain cannot cause the disease and S strain is activated, still the mice was dead. So what was the conclusion of this experiment? Griffith concluded that there is some transforming principle. There is some transforming principle which is added to the medium by the S strain and taken up from the medium by the R strain, because R strain is naturally a competent strain. That means it can easily accept the foreign molecules. So when this R strain took up something from the medium, it transformed into the S strain and caused the disease. But Griffith did not have any idea that what is the biochemical nature of this transforming principle. He just said that there is something which is added to the medium by S3 strain and something which is taken up from the medium by the R2 strain. What is this something? He called this something as transforming principle. He did not have any idea whether this is DNA, RNA or protein. So, in the time period between 1933 to 1944, it was assumed that protein was the genetic material. Between 1933 and 1944, protein was considered as the genetic material. But something changed in the year 1944. And what's changed? Let's discuss. In the year 1944, there was experiment, that is MacLeod, McCarthy and Avery's experiment. They liked the experiment of Griffith and repeated the experiment in lab conditions. That means they repeated the experiment in vitro conditions. They did not use mice for their experiment, but used the in vitro conditions. Now what they did? Let's understand. They took the heat-killed strain and extracted, isolated the biochemicals of the S strain. Okay? Now this test tube you are seeing here, this contains the biochemicals extracted or isolated from the S strain. So this is a mixture. This mixture contains DNA, RNA, protein, lipids, sugar, etc. But they extracted out lipids and sugars deliberately from this mixture. Now, this mixture contains mainly DNA, RNA and protein. Now, what they did? They took 3 test tubes, as you can see in the experiment. What is there in these test tubes? The test tubes has the R strain in it. So these tubes have R strains in them. When we add this whole mixture to the test tube, that means we are adding DNA, RNA and protein of the S strain which we have just extracted. So when we added this mixture of biochemicals into the first test tube and along with this mixture, we added the enzyme proteinase, or you can say we added the enzyme protease. Now, what is the function of enzyme protease? It degrades the protein. So, in this first tube, we have a mixture, but after adding the enzyme protease, it degraded the protein. Even after the degradation of protein, you can see here in the test tube that the R cells were getting transformed into the S cells. That means transformation was still there. So what does this prove? That protein is not the transforming principle. It does not have the capacity to transform because even in its absence, the transformation is going on. Then in the second test tube, we again have the R bacteria and we again are adding the mixture of biochemicals, but now along with it, we are adding the enzyme RNase. What does RNase do? RNase degrades the RNA. But even after the degradation of RNA, there is still the transformation happening. That means RNA is also not the transforming principle, because even in the absence of RNA, the transformation is still happening. Then they took the third test tube, again added the mixture, and along with it, they added the enzyme DNase, and DNase will degrade the DNA. After the degradation of DNA, the transformation stopped. That means the transforming principle Griffith talked about is nothing but DNA. Because in the absence of DNA, transformation stops and R strain cannot convert into the S strain. So with this experiment, the topic of the discussion changed. Up till now, everybody used to think that protein is the genetic material, but now the topic of the discussion is changed. Now there is the new biochemical in the picture, and that is DNA. So MacLeod, McCarthy, and Avery told that the transforming principle of Griffith is nothing but DNA. So hence, they were the first ones to suggest that DNA is the genetic material. But nobody accepted their explanation, because since a long time everybody assumed that protein is the genetic material, and now everybody was not ready to accept the fact that DNA is the genetic material. But now something will come in the picture that will change the whole thing about the confusion regarding the genetic material, and that experiment is the Hershey and Chase experiment, which came in the year 1952. In the year 1952, it was stamped, it was proved, it was declared that DNA is the genetic material. The discussion is stopped, period. We all accept now that DNA is the genetic material. So there is a line in the NCERT that this experiment, Hershey and Chase experiment, is the unequivocal proof. What is the meaning of unequivocal proof? That means it cannot be challenged anymore. Everybody will have to accept it that DNA is the genetic material. So question can come, a direct MCQ can come, who gave the unequivocal proof that DNA is the genetic material? The answer would be Hershey and Chase experiment. Okay, so let's discuss what are the materials he took in his experiment. He took the bacteria E. coli. He took the bacteria E. coli and he took bacteriophage. You know what is bacteriophage? It is a virus that infects bacteria. So we have two materials for this experiment, E. coli, bacteria, and bacteriophage, that is the virus. To understand this experiment, we need a quick brief regarding the structure of the virus. So let's understand a basic structure of virus. Virus is nothing, it is just a protein plus genetic material. Genetic material is surrounded by protein coat. So this is virus. It has the protein coat. The black structure here you can see in the image represents the protein coat, and the blue structure you are seeing here is the DNA. So a virus is basically composed of two things, that is protein coat and the DNA. And the bacteriophage we'll be using here, you must have heard many names of bacteriophages like lambda phage, T1 phage, T2 phage, T3 phage. So they have this sort of structure, and the genetic material of this bacteriophage is generally a double-stranded linear DNA. A double-stranded linear DNA. Okay? So let's start with the experiment. So to understand the experiment, first of all, let's understand the term, what is radioactive labeling? In this experiment, the protein coat of the bacteriophage will be radioactively labeled and the DNA of the bacteriophage will be radioactively labeled. What is the meaning of radioactive labeling? When we insert some radioactive substance into the protein coat or the DNA, that means radioactive labeling. What is the purpose of radioactive labeling? When we radioactively label some substance, then it fluoresces. It shows fluorescence in the presence of X-rays. That means they will say, I'm present here, I'm present here. You will have to do nothing, but they will talk about themselves that we are present here. So why radioactive labeling is done? To detect the presence, because they will show fluorescence. So for that sort, what they did, they took two mediums. Okay, one medium contained sulfate and the another medium contained phosphate. Okay, sulfate and phosphate. But the sulfate in this medium is not the normal one, but the radioactive isotope of sulfur, that is S35. And the phosphate in this medium is not the normal one, but the radioactive isotope of phosphorus, that is P32. Okay, when they grew the bacteriophage in the medium S35, in the medium containing sulfate having S35 and the medium containing phosphate having P32, then what happened? We are now with the bacteriophage whose protein coat is radioactively labeled. That means for this you have to understand one thing, beta, that sulfur is present in the protein and phosphorus is present in the DNA. We know that DNA has phosphate group at its 5' end. So phosphorus in DNA and sulfur in protein coat. Okay, so when we grow the bacteriophage in this medium having S35 Wallah sulfate, then that means the protein coat of the bacteriophage will be radioactively labeled because sulfur is present in the protein. So I am denoting the radioactively labeled condition with this pink color. So here we have got the bacteriophage whose protein coat is radioactively labeled, and here we have got the bacteriophage whose DNA is radioactively labeled. So now we have two types of bacteriophages, one with radioactively labeled protein coat and the other with radioactively labeled DNA. Okay, let's move forward now. Let's start with the experiment now. But before starting the experiment, we should understand some points. First point, we know that when virus infects the bacteria, it take over the machinery of bacteria and divide itself, make many copies, then rupture the bacteria and comes out. That means it has to make colonies of itself inside the bacterial cell. So obviously a virus during infection will inject its genetic material into the bacteria because only the genetic material has the whole information to form a new organism. So virus, when it will go inside the bacteria, it has to form a copies of itself. So obviously it will prefer to inject the genetic material into the bacteria so that it can form new types of its own self. So one thing we know even before the start of experiment, that whatever the virus will inject into the E. coli will be the genetic material, right? This is what we know even before the experiment. Then, what are the steps which would be involved in the experiment? First, infection. During infection, the virus will inject its genetic material into the E. coli because that is preferable. The second step is blending. What we do in the blending, that some part of the virus will go into the E. coli and some part of it will not go into the E. coli cell, but it would be attached to the surface. We want this part of virus to be detached from the surface, and for that sort, we put this mixture on shakers and vigorously shake it so that the part of virus which is not gone inside the E. coli would be detached from the E. coli. And the third step is centrifugation. What we do during centrifugation, we put this whole mixture into small tubes called as Eppendorf tubes, and then we put that Eppendorf tube into the machine called as centrifuge machine, and we rotate it at high RPM. What is the result of centrifugation? The result of centrifugation is this. You will get precipitate at the bottom and you will get supernatant, that is that liquid part above the precipitate. So this is the result of centrifugation, formation of precipitate, and you will have a supernatant. So these are the steps of the experiment. Now let's understand what was the experiment. Let's divide the experiment into two parts. In the first part, we can see that E. coli is being infected with the virus having radioactively labeled protein coat. And in the second set of experiment, the E. coli is being infected with the bacteriophage having radioactively labeled DNA. Okay? First step, infection is done. Whatever they have to inject into the E. coli, they have injected. Then blending. The part of virus which is not gone into the E. coli, it is separated. And then last, we did the centrifugation. And then in both the cases, we got precipitate and the supernatant. Okay? Let's go forward. So now the question is, what is there in the precipitate and what is there in the supernatant after centrifugation is finished? In the precipitate, there would be the heavier part. That means, in the precipitate, there would be E. coli cell plus the part of virus which is injected into the E. coli. That will precipitate. And the part of virus which is not injected into the E. coli would be present in the supernatant. So what we know right now, that supernatant will have the part of virus which is not injected into the E. coli, and the precipitate would be having E. coli plus the part of virus injected into the E. coli. Okay? So let's discuss the conclusion now. What is the conclusion of the experiment? So now we have two Eppendorf tubes. One from the experiment in which the bacteriophage was having S35 in its protein coat, and the second Eppendorf tube in which the bacteriophage was having P32 in its DNA. So what is the conclusion now? This is important. Understand it. In this Eppendorf tube, the fluorescence was present in the supernatant. The fluorescence was present in the supernatant. And the presence of fluorescence in the supernatant depicts that supernatant has that part of virus which is radioactively labeled. And we know what part of virus was radioactively labeled in this experiment. Protein coat. So the fluorescence in the supernatant depicts that in the supernatant, the protein coat of the virus is present. That means protein coat is not injected into the E. coli because supernatant contains the part of virus which is not injected into the E. coli. So what does that depict? That protein is not the genetic material because virus has not injected its protein coat into the E. coli. That is why it is present in the supernatant, and that is why it is showing the fluorescence. In the second set of experiment, the fluorescence is present. The fluorescence is present in the precipitate. That means P32 is present in the precipitate. That means DNA of virus is present in the precipitate. And precipitate used to contain E. coli plus part of virus, which is injected into the E. coli. That means that DNA is the material which is injected into the E. coli by the virus. And this proves that DNA is the genetic material. And after this experiment, it was stamped that DNA is the genetic material. So now the search, the race for finding that who is the genetic material is finished. Now everybody, me, you and I, everyone knows that DNA is the genetic material. So now let's move on to the other part of the chapter. Now we know that DNA is the genetic material. So let's start with the structure of DNA. As you all know that DNA was first termed as nuclein. It was first termed as nuclein by the scientist Friedrich Mischer. And we know that DNA is an acidic substance and it is negatively charged. It is negatively charged. Okay? DNA is a polymer, and it is a polymer of the repeating units, that is deoxyribonucleotides. Deoxyribonucleotides. And what are they? Let's discuss. Let's change the color, make it more colorful. DATP, DCTP, DGTP, and DTTP. These are the nucleotides of DNA. Deoxyadenosine triphosphate, deoxycytidine triphosphate, deoxyguanosine triphosphate, and deoxythymidine triphosphate. These are the nucleotides of DNA. So let's understand what is the basic structure of a nucleotide. A nucleotide contains three parts. First, a pentose sugar, and in case of DNA, this pentose sugar is a deoxyribose sugar. Second, it contains a nitrogenous base, and third, it contains a phosphate group. So these are the three constituents of the nucleotide of DNA. Nitrogenous bases further are of two types, that is purines and pyrimidines. Purines are of three types, adenine, guanine, uracil. Uracil is present in RNA and adenine and guanine are present in DNA. Pyrimidines are of two types, cytosine, thymine. Okay, so let's understand the diagram of the nucleotide of DNA. In the center, you can see a deoxyribose sugar. This is in the center, it is a deoxyribose sugar. Now, why it is called as deoxy? Because you can see it, let me number the carbons. This is 1' carbon, 2' carbon, 3' carbon, 4' carbon, and 5' carbon. 5' carbon of the sugar is present outside the ring. So at the 2' position beta, there is only hydrogen, not the OH group. That is why it is called as deoxyribose sugar. At the 1' carbon, of the sugar, the nitrogenous base is associated, and at the 5' carbon of the sugar, the phosphate group is associated. So what you can say in this condition that we can also use a term that is nucleoside. Nucleoside is equal to sugar plus nitrogenous base. As soon as at the 1' carbon of the sugar, the nitrogenous base get attached, it becomes nucleoside. And as soon as phosphate attaches at the 5' carbon, it becomes nucleotide. And in the case of DNA, the most appropriate word would be deoxynucleotide. Okay, so this is the basic structure. Now understand the bonds present inside one single nucleotide. The bond between the sugar and nitrogenous base is N-glycosidic bond. Is N-glycosidic bond, and the bond present between sugar and the phosphate group is called as the phosphoester linkage. Phosphoester linkage. It's phosphoester, not phosphodiester. Okay, so within one single nucleotide, you have two types of bond. That is N-glycosidic bond within single nucleotide. We have N-glycosidic bond and phosphoester bond. Okay, between two nucleotides, we have phosphodiester bond. And we will do many questions associated with this topic in the MCQ section. So this is the diagram of NCERT, as you all must know. So let's understand what happens when nucleotide attaches and forms a polynucleotide chain. So beta, this is the sugar. As you can see, this is the sugar. Okay, this is the 1' carbon of the sugar, and this is the 5' carbon of the sugar. At the 5' carbon, phosphate group, 1' and the nitrogenous base. This is the N-glycosidic linkage, and this is the phosphoester linkage. Okay, N-glycosidic linkage and the phosphoester linkage. Now comes the phosphodiester bond between two nucleotides. This is one single nucleotide and this is the other nucleotide. This is the other nucleotide, and between two nucleotides we have phosphodiester bond. Phosphodiester bond. Okay, so many question comes regarding the calculation of number of phosphodiester bonds in a particular DNA. So let's deduce a general formula for calculating the number of phosphodiester bonds. As we all know, beta, we can have four types of condition in the DNA. That means the DNA can be double-stranded linear. It can be double-stranded linear. The DNA can be single-stranded linear, or it can be double-stranded circular, or it can be single-stranded circular. We can have these 4 conditions in the DNA: double-stranded linear, single-stranded linear, double-stranded circular, and single-stranded circular. So now we want to calculate the number of phosphodiester bonds. To calculate the number of phosphodiester bonds, let's have a general formula. In this condition, beta, double-stranded circular condition, we will calculate the number of phosphodiester bond by the formula. Number of phosphodiester bonds will be equal to number of nucleotides minus 2. Number of nucleotides minus 2. In case of this condition, when the DNA is single-stranded linear, then the number of nucleotides will be equal to, number of phosphodiester bonds will be equal to number of nucleotides minus 1. In case of either double-stranded circular DNA or single-stranded circular DNA, in both the cases, number of phosphodiester bonds will be equal to number of nucleotides. Okay? So, in the DNA having the double-stranded linear condition, number of phosphodiester bonds is equal to number of nucleotides minus 2. In this condition, single-stranded linear DNA, number of nucleotides minus 1. And in this condition, it would be equal to number of nucleotides. But be very careful that what is given in the question. They have given the number of base pairs, or they have given the number of nucleotides or bases. Suppose, for instance, there is a question which says that there is a double-stranded linear DNA and that double-stranded linear DNA has one, you can say, chalo, let's take 100 base pairs. 100 base pairs. So you'll have to convert these base pairs first into nucleotides. Please don't do that. You directly say 100 minus 2 comes out to be 98. No. Base pairs needs to be first converted into nucleotides. So one more thing, that if we are talking about the double-stranded condition, then the nucleotides would be present in the pairs. That means in the double-stranded condition, we would be having base pairs. And in the single-stranded condition, we would be having only nucleotides or bases, no base pairs. So here it is given that there are 100 base pairs. So first, convert it into number of nucleotides, that is 100 into 2, because one pair contains 2 nucleotides. So 100 base pairs will be having 200 nucleotides. Or you can say 200 bases. 200 nucleotides or 200 bases, not base pairs. And now we can calculate the number of phosphodiester bonds. Number of phosphodiester bonds in this case will be equal to number of phosphodiester bonds will be equal to 200 minus 2. That comes out to be 198. Okay, so this is how you can directly apply these formulas to calculate the number of phosphodiester bonds. And what will be the number of glycosidic linkages? Number of glycosidic linkages will be equal to number of nucleotides. Always, because one nucleotide contains one glycosidic linkage. But phosphodiester bond is present between two nucleotides. So number of n glycosidic linkages will always be equal to number of the nucleotides. And be careful, you have to first convert the base pairs into the nucleotides. And if the question is directly saying that nucleotides, then there is no need for conversion. Okay, let's move further. Yes, this is the important data in NCERT beta. We can calculate the length of DNA either in the form of base pairs if it is a double-stranded condition, or in the form of nucleotides or bases if there is single-stranded condition. So let's learn these data along with me because they are important. First organism given here is Phy174, and Phy174 is a bacteriophage. That means it is a virus. And what sort of DNA it has? It has a single-stranded circular DNA. That means this type. That means it would not be having base pairs. It would be having only nucleotides or bases. And the number of nucleotides are 5386 nucleotides or bases. Repeat with me, 5386 nucleotides, not base pairs. Then the second organism given here is lambda-phage. This is also a bacteriophage or virus which has double-stranded linear DNA. That means this type. And it has 48502. Learn with me, 48502 base pairs. Base pairs, not nucleotides or bases. Then E. coli. E. coli is a bacteria having double-stranded circular DNA. Double-stranded circular DNA, that means this kind of condition. And it has 4.6 into 10 raised to the power 6 base pairs. And the last is human genome. What is the meaning of genome? Genome represents the haploid content. Genome represents the haploid content. That means the number of chromosomes in our sperm or ovum are 23 and the DNA content present in it has this condition. That is 3.3 into 10 raised to the power 9 base pairs. Okay, 3.3 into 10 raised to the power 9. If it is asked that what are the number of base pairs in a human diploid cell, then in a human diploid cell or any mammalian diploid cell, the number of base pairs would be 2 into 3.3 into 10 raised to the power 9. That comes out to be 6.6 into 10 raised to the power 9 base pairs. Okay, so in a human diploid cell, the number of base pairs would be 6.6 into 10 raised to the power 9, and in the haploid cell it would be 3.3 into 10 raised to the power 9. And you can carefully read the question whether the number of base pairs are asked or the number of nucleotides are asked. If the question says that what is the number of nucleotides in a sperm of humans, so humans have 3.3 into 10 raised to the power 9 base pairs. So the number of nucleotides in the sperm would be 6.6 into 10 raised to the power 9. But if the question is saying that calculate the number of base pairs in the diploid cell, that would be 6.6 into 10 raised to the power 9. And what would be the number of nucleotides in the human diploid cell? 2 into 6.6 into 10 raised to the power 9. Okay? So read the question carefully. So now we know what is the structure of DNA, and nucleotides are its monomeric units. Within a single nucleotide, you have N-glycosidic linkage, and between the two nucleotides, you have phosphodiester bond. Now, to understand the structure of DNA, let's discuss the Watson and Crick model, which came in the year 1953. That means just after the discovery that DNA is the genetic material in 1952 by Hershey and Chase, Watson and Crick, just after 1 year, provided the detailed structure of DNA. But the model of Watson and Crick is not independent. It is based on the work of two scientists. That is Wilkins and Franklin, and the other one is Erwin Chargaff. The Watson and Crick model is based on these two works. Let's discuss what is Wilkins and Franklin model. It is a model based on X-ray diffraction studies. What is it? They took a DNA solution and exposed this solution with X-rays, and what they got in the photographic film X-ray photographic film, they got this kind of picture of DNA. That means, who were the first ones who gave the idea that DNA has a double helical structure? They were Wilkins and Franklin with the help of X-ray crystallographic studies. So they suggested that DNA has double helical structure. Okay, I hope my writing is clear. Yes, Arvind Chargaff, what did they do? They did the base composition studies. That means they tried to understand that DNA is composed of what bases and in what proportion. In what proportion mainly. So they did the base composition studies, and he found out that adenine is always equal to thymine. Adenine is always equal to thymine. That means adenine and thymine are always equimolar. That means A and T are always equimolar in the DNA. Okay? And then they found out that C is always equal to G. That means the ratio A by T is always equals to 1, and the ratio C by G is always equal to 1. And they told that the total content of purines in the DNA is always equal to the total content of pyrimidines. That means A plus G is always equal to C plus T. That means A plus G upon C plus T will always come out to be 1. But the ratio A plus T upon C plus G is not always equal to 1. That is different for different species. For prokaryotes, this ratio is less than 1, and in case of eukaryotes, this ratio is more than 1. And what does that suggest? That means that prokaryotes have GC-rich DNA and eukaryotes have AT-rich DNA. Okay? Prokaryotes have GC-rich DNA and eukaryotes have AT-rich DNA. Okay. Now, what do we know? We know two things. And what are they? With the help of the Wilkins and Franklin experiment, we know that DNA has a double helical structure. And with the help of Chargaff's experiment, we know that A is always equal to T and C is always equal to G. Now, using these two experiments came the Watson and Crick model. So let's discuss the points of Watson and Crick model quickly. Yes, NCERT says the hallmark of Watson and Crick model is complementary base pairing. Complementary base pairing. Hallmark means this is the most important point of the Watson and Crick model, that is complementary base pairing. That means that A will always pair up with T and C will always pair up with G. This is complementary base pairing. And from where this idea came from? Obviously, Chargaff rule, because Chargaff said A is always equal to T. Why A is always equal to T? Because A is always found with T. And why C is equal to G? Because C is always found with G. Because they form pairs with each other. And this is called as complementary base pairing. And between the two, these two nitrogenous bases present on the two strands, they have hydrogen bonds. Between A and T, there are 2 hydrogen bonds, and between C and G, there are 3 hydrogen bonds. Okay? Now, other important points of Watson and Crick model are that because purine will always be paired up with pyrimidine. This is the line of NCERT. Listen carefully. Because purine is always paired up with pyrimidine, so this is the reason why the distance between the two strands, dhyan dena beta, distance between the two strands, listen carefully, is always uniform. Okay, so this question can come directly. What is the reason that the distance between 2 strands of DNA is nearly uniform? So you will say that purines always pair up with pyrimidines because of the complementary base pairing, A with T and C with G. Okay. Now again, another type of question which is very common, that what is the reason that this helical structure of DNA is stabilized? What is the reason behind the stability of the helical structure of DNA? There are 2 reasons. First, the plane of one base pair stack over the other. The plane of one base pair stack over the other. And the second reason is the hydrogen bonds between the 2 strands. Okay, so these are very important points. What generates the uniform distance between the 2 strands of DNA? Complementary base pairing, that is purine always pairs up with pyrimidine. What is the reason behind the stability of helical structure of DNA? First, plane of one base pair stack over the other, and second, hydrogen bonding. Okay. Now, some additional points of Watson and Crick model. It says that DNA is a double helical structure having 2 strands. This is one polynucleotide chain. This is one polynucleotide chain. And this is the other polynucleotide chain. So in the structure of DNA, we have 2 polynucleotide chains. This is one strand and this is the other strand of DNA. So DNA has 2 strands or 2 polynucleotide chains. And these 2 chains, these 2 strands are anti-parallel to each other. 2 strands are anti-parallel to each other. Anti-parallel means the polarity of one strand is 5' to 3' and the polarity of other strand is 3' to 5'. And how do we read the polarity? In the left to right direction. Remember, in the left to right direction, 5' to 3', 3' to 5'. They are anti-parallel to each other, and the backbone of the polynucleotide chain, is formed by sugar and phosphate. The backbone is formed by sugar and phosphate, and the nitrogenous bases, they are present towards the inner side. Okay? So these are some important points of the Watson and Crick model, and this is the diagram of DNA which explains the double-stranded structure of DNA. So this is the one polynucleotide chain or the one strand, and this is the other polynucleotide chain or the other strand. And you can see it here that this is the N-glycosidic linkage, this is the phosphoester linkage, this is the phosphodiester linkage, and between the two strands of DNA, this is one strand, this is other strand, and between them we have hydrogen bonds. So these are the hydrogen bonds. These are the hydrogen bonds. And these hydrogen bonds are responsible for the stability of the helical structure. Okay? So let's discuss some dimensions of the DNA also. In the DNA helical structure, one helix or one pitch length, one helix, the one complete turn has 10 base pairs, and the pitch length is 3.4 nanometer. 3.4 nanometer. So remember that helix or pitch length is equal to 3.4 nanometer. One helix or one pitch length is 3.4 nanometer. And one helix contains 10 base pairs. That means distance between one base pair is equal to 0.34 nanometer. These dimensions are important. And distance between two strands, or you can say the helical diameter. The helical diameter is equal to 2 nanometer. You have to learn these dimensions in one unit. Just learn them in nanometer. One helix means one complete turn that contains 10 base pair. The length of one helix is 3.4 nanometer. Do not learn it in angstrom. Learn it in nanometer, and then you can convert it in angstrom if it is given in angstrom in the question. So learn all the dimensions in nanometer. One pitch length, 3.4 nanometer, distance between two base pair, 0.34 nanometer, and the helical diameter. Helical diameter means this thing, the distance between the two strands. This is the helical diameter, that is 2 nanometer. So these are the important dimensions. And this is the Watson and Crick model. So now we know that what is the genetic material, that is DNA, and we have understood the structure of DNA thoroughly. Now let's discuss the theory, that is central dogma theory, which is given by Francis Crick. This theory says that what is the flow of information in the nature? The flow of information is DNA to RNA and RNA to protein. DNA is the storehouse of the genetic information. But for the transmission of genetic information, we need RNA. And finally, for the expression, we have blue eyes, or we have dark skin color, or we have black color. This is due to the protein formed by ultimately by the DNA, which codes for the information. So the information is passed on to RNA, and then RNA to protein, and finally the protein is responsible for the phenotype or for the expression. This is central dogma theory. So DNA can replicate itself. That means when it says that DNA to DNA, it would be replication. This is the diagram of NCERT, that when DNA forms a copy of itself, it is replication. Then DNA transfer its information to RNA, then that is transcription, and RNA transfer its information to protein, that is translation. Okay, and one more thing is given in NCERT, and that is beta. Yeah, that is reverse central dogma. What is reverse central dogma? We know that when DNA forms DNA, it is replication. When DNA forms RNA, it is transcription. But sometimes in some retroviruses, RNA can also form DNA, and this is reverse transcription. This is reverse transcription. So, reverse central dogma theory says that in some cases, in some retroviruses like HIV, that is AIDS virus, it can be seen that the flow of information can be reversed, that is from RNA to DNA. Okay, this is reverse central dogma and can be seen in some retroviruses because they have a specific enzyme, that is reverse transcriptase for this sort. Now, let's move on to the packaging of DNA. Yes, the packaging of DNA. Now we know what is DNA. Now we know everything about DNA, but what we know is that the length of DNA is pretty big. If we discuss the length of DNA in a diploid mammalian cell, then we know that the distance between two base pairs is 0.34 nanometer. That is 0.34 into 10 raised to the power, it should be minus 9, and this is 6.6 into 10 raised to the power 9. Okay, total number of base pairs 6.6 into 10 raised to the power 9 and 0.34 into 10 raised to the power minus 9 meter. So the distance comes out to be 2.2 meters. And in E. coli, in bacteria, the length of the DNA comes out to be approximately 1.36 millimeter. So learn with me. In human diploid cell, the length of DNA is 2.2 meters. In E. coli, the length of DNA is 1.36 millimeter. So now we know that the diameter of nucleus is of the order of 10 raised to the power minus 6 meters. So how is this possible that the length of DNA, which is 2.2 meters, can be adjusted in a nucleus of such a small diameter? That means it needs supercoiling, it needs condensation, and that is called as the packaging of DNA. The packaging in case of prokaryotes is not as complex as in case of eukaryotes because prokaryotes does not have nucleus, but their DNA is directly present in the cytoplasm and called as nucleoid. This is given in NCERT, called as nucleoid. So in case of E. coli, the packaging is pretty easy, but the cell of E. coli is itself of the order of 10 raised to the power minus 6 meter. So it also needs packaging, but not as complex as in case of eukaryotes. So in E. coli, for packaging, in E. coli, which has nucleoid, what is nucleoid? You can say that the DNA which is present directly into the cytoplasm, that region of cytoplasm is nucleoid. Directly in cytoplasm, because this is not surrounded by nuclear membrane. So for packaging of DNA in E. coli, for packaging, histone proteins are not present. You must have heard of special proteins called as histone proteins, which are present in eukaryotes, but in E. coli or in bacteria, for packaging, they have another type of proteins called as polyamines. Histone proteins absent. Polyamines are also basic proteins. That means they are composed of basic amino acids and they also are positively charged. But in case of E. coli, histone proteins are absent. Okay, so in place of histone proteins, they have polyamines, which are responsible for the supercoiling or packaging of DNA in case of bacteria. But it is complex in case of eukaryotes. Let's discuss that now. For the packaging of DNA in case of eukaryotes, we have special proteins, and those are histone proteins. Now, what are the characteristics of histone proteins? They are positively charged. They are rich in the amino acids, that is lysine and arginine. This is an important point. Lysine and arginine are basic amino acids. That is why histone proteins are basic proteins. There are 5 types of histone proteins which are found, and they are H1, H2A, H2B, H3, and H4. These are the 5 types of histone proteins found in eukaryotes. Now let's discuss that in what way they are responsible for the packaging. First of all, let's understand the formation of NU body. Formation of NU body, or you can say histone octamer. NU body or histone octamer. Okay. So understand like that NU body or histone octamer is like a ball, and on this ball we have to wrap the DNA and try to condense it. So histone octamer is composed of 2 molecules each of H2A, H2B, H3, and H4. That means how many balls? 8 balls. 2 of H2A, 2 of H2B, 2 of H3, and 2 of H4. These 8 balls will combine and they form a octamer, histone octamer, also called as NU body, on which we have to wrap the DNA. So understand that this is a ball which is called as histone octamer, and on this ball we will wrap the DNA. Okay. This DNA wrapped over the nucleosome is called as the core DNA. So we are wrapping this DNA on the nucleosome. The DNA takes 1.75 turn on this ball. That means 1 turn, second turn is not completed. It takes 1.75 turn on the DNA, and then we will take the another ball and will wrap the DNA. So many balls, we will take many balls and will wrap the DNA. 1, 2, 3, 4. So between 2 nucleosomes, there would be some part of DNA which would be present, and this is called as the linker DNA. So 1 nucleosome is equal to the ball, that is histone octamer, the DNA wrapped over it, that is core DNA, and the linker DNA. Okay, so 1 nucleosome contains NU body, that is histone octamer, plus core DNA plus linker DNA. This is 1 nucleosome. And 1 nucleosome, this is very important. one nucleosome contains 200 base pairs of DNA. This is very important. One nucleosome contains 200 base pairs of DNA. It covers 200 base pairs of DNA. Okay. So let's understand this with a picture. This is the diagram of a nucleosome. At the center, you can see 8 balls, that is histone octamer or NU body, over which you can see the DNA wrapped over. This is the core DNA, this is the linker DNA, and this is the H1 histone. H1 histone do not participate in the formation of histone octamer, but it acts like a clamp which holds the DNA on this ball. Okay, so remember this point. H1 histone do not participate in the formation of octamer, but act as a clamp. So it acts as a clamp. Okay, and this DNA takes how many turns? It takes 1.75 or 1/3/4 turn on NU body. So this is a nucleosome. So what would be the appearance of nucleosome? The appearance of nucleosome will be beads on string. Nucleosome will appear as beads on string. That means there is a one long thread on which many beads are present. This is a nucleosome. So let's understand how packaging is done. First of all, DNA condenses to form nucleosome. Then nucleosome further condenses to form chromatin fibers, and ultimately it super condenses to form chromosome. Chromosomes are not seen in a resting cell, but the fully condensed form of DNA is seen in a dividing cell, and that too at metaphase. So nucleosome condenses to form chromatin fibers, then chromosomes, and chromosomes are seen at the metaphase. There are some other type of proteins which are involved in packaging, non-histone chromosomal proteins. These proteins are acidic in nature. These proteins are acidic in nature and they are used in higher level of packaging. Okay. So after packaging, we are here with two terms, euchromatin and heterochromatin. During packaging, some parts of DNA are densely condensed, and those parts are called as heterochromatin, and some parts are loosely packed or loosely condensed. Those parts are called as euchromatin. If we stain this chromatin material, then heterochromatin would be darkly stained and euchromatin would be lightly stained. Here you can see that euchromatin is transcriptionally active and heterochromatin is transcriptionally inactive. That means the densely condensed DNA, on the densely condensed DNA, RNA will not be formed. That means the heterochromatic part of DNA will not be expressed, but the euchromatic part of DNA will be expressed. Now let's compare DNA versus RNA. DNA usually is double-stranded. DNA usually is double-stranded, as you can see, and RNA is usually a single-stranded molecule. Second difference, DNA contains deoxyribose sugar. Deoxyribose sugar, you know that at 2' carbon, there is hydrogen and not the OH group. But in the RNA, we have ribose sugar, that is, there is OH group at the 2' carbon. Then DNA has the nitrogenous base thymine, but the RNA has the nitrogenous base uracil in place of thymine. Thymine can also be called as 5-methyl uracil. Because what is the difference in the structure of thymine and uracil? In uracil, there is no methyl group at the fifth position. But in case of thymine, there is methyl group at the 5th position. That means if you put a methyl group at the 5th position in the structure of uracil, it becomes thymine. So there is a question which comes that presence of uracil is normal or abnormal in DNA? Obviously abnormal. But the presence of 5-methyl uracil is normal in DNA because 5-methyl uracil is the another name of thymine. Then what was the first genetic material on Earth? The first genetic material on Earth was RNA. And slowly and gradually, DNA has evolved from RNA to take over the position of the genetic material. DNA can only act as genetic material and is responsible for the storage of genetic information. It is responsible for the storage of genetic material. But RNA, it used to act as genetic material, but now it also acts as a catalyst. Catalyst means it also acts as a enzyme. There are certain ribozymes which we will be dealing in this chapter in the later part. Peptidyl transferase enzyme, it's a type of ribozyme where rRNA behaves as ribozyme. So RNA can also behave as enzyme, and RNA has structural role also. We know that ribosomes are formed from rRNA. So RNA has the structural role also. It can act as catalyst also, and it used to act as the genetic material. So if you are saying that who is responsible for the storage of genetic information? DNA. For the transmission of genetic information? RNA. So RNA is responsible for the transmission of genetic information. Okay. Now, we have discussed the biochemicals involved in this chapter. Now let's discuss the very important process, that is replication of DNA. What is the meaning of replication? When DNA, one copy of DNA makes its 2 copies. That is replication of DNA. But before that, let's discuss about the nature of replication. The nature of DNA replication is semi-conservative. What is the meaning of semi-conservative? Semi-conservative means that DNA is a double-stranded structure. DNA is a double-stranded structure, and during replication, it will form 2 copies of itself. 2 copies of itself, but when it will make 2 copies of itself, then these 2 new DNA molecules, they will be having one strand, that is the parental strand, but the other strand would be newly formed using the medium. That means the nature of DNA replication is semi-conservative. One strand would be parental and one strand would be new. So this is the parental strand and this is the new strand. So this is the semi-conservative nature. The semi-conservative nature of replication was suggested by Watson and Crick. Suggested. But it was proved that DNA replication has semi-conservative nature. In prokaryotes, it was proved by Messelson and Stoll, and in case of eukaryotes, it was proved by Taylor et al. Messelson and Stahl proved it in using the organism E. coli. And Taylor et al. Proved it using the organism Visia faba. That is broadwheels. Now let's discuss the Messelson and Stahl experiment. What did they do? To start with the experiment, let's understand that E. coli has double-stranded circular DNA. It has double-stranded circular DNA, okay? And the nitrogen which is present in both these strands of E. coli is of N14, the normal nitrogen. So normally, in normal case, what we can denote is that the DNA of this E. coli is 14-14 DNA. Okay? 14-14 means that in one strand, all the nitrogen is N14 and in the other strand also, all the nitrogen is N14. Okay? So, what did they do to start with the experiment? They grew E. coli having this 14-14 DNA, on the medium N15H4Cl. N15H4Cl. Now, what is this 15 here? This 15 is the heavy isotope of nitrogen, not the radioactive isotope. Remember, not the radioactive isotope, but the heavy isotope of nitrogen. So, ammonium chloride, N15 ammonium chloride. And they grew the E. coli in this medium for several generations. And after growing this for several generations, what they obtained the E. coli in which all the nitrogen was of N15. We started with the 14-14 DNA, then the medium taken was N15H4Cl, and then after growing it for several generations, the DNA here is 15-15 DNA. 15-15 DNA. That means now we have the E. coli having all the nitrogen in both the strands N15. Okay. Now, let's start the experiment. Till now, what we have done? Till now, we've just made a bacteria having all the nitrogen 15-1. But now we'll actually start the experiment. Now we'll take the medium, and this medium is N14H4Cl. N14H4Cl, okay? And in this medium, we will, now the medium, remember, now the medium is N14H4Cl, and the E. coli present in it having the DNA 15-15. Okay? So, after 20 minutes of division, we'll get the first generation, and the first generation will be like this. Just a second. Let's use the proper colors so that it will easily understand. After first generation, we will be having this condition. One of these strands would be parental, that is 15, 15, and one of these strands would be non-parental, that is 14, 14. So what we have got in the first generation, we have got two DNA molecules, and those two DNA molecules, they are 15, 14, 15, 14. That means hybrids in this first generation. Okay. Then after further 20 minutes, what we'll get 4 DNA molecules, and those 4 DNA molecules, this DNA molecule would be forming 15, and one would be taken from the medium, that is 14, and other strand is 14, and one will be taken from the medium, that is 14. So this one DNA molecule will form 2 DNA molecules, that is 15, 14 and 14, 14. 15, 14 and 14, 14. Likewise, this lower one will also form 15, 14 and 14, 14. Are you getting my point? That means that in first generation we have got the hybrids. 15, 14 are the hybrids, 15, 15 are the pure heavy, and 14, 14 are the pure light. So in the first generation we had only hybrids. But in the second generation we have, one second. Yeah. So in the second generation we would be having this. You can see here, Vita. Just a second. Yeah. So in the second generation, we are having pure light and hybrids. There are no pure heavy. So keep this in mind. Pure. What type of pures would be formed would be dependent on the type of medium you are taking. If you are taking the medium N14, then there will always be pure light and hybrids. If you are taking the medium N15, then there would be hybrids and pure heavy. So in this case, there are pure light and hybrids. Okay. So how this experiment make us understand that replication is a semi-conservative process after we do a density-based centrifugation, that is cesium chloride-based centrifugation. So after the CSCl-based centrifugation, that means density-based centrifugation, what we get in the first generation, we got only one band. In the second generation, after density based centrifugation with the help of cesium chloride, CSCl, we got 2 bands. Here the band was only of 15, 14, that is only of hybrids, but here the band is of pure light, 14, 14, and the hybrids 15, 14. These are pure light and these are hybrids. That means this formation of 2 bands in the second generation after centrifugation proves that DNA replication is semi-conservative. If it would have been fully conservative, then no matter for how many generations the experiment goes on, the band would have been only 1. The presence of 2 bands prove that replication is semi-conservative. One of the strand comes from the medium, synthesized with the help of medium, and the other strand is the parental strand. The question related to this topic we'll be doing in the MCQ section, the detailed question that what will happen after first generation, what will happen after second generation. Now, let's move on to the replication of DNA. Replication of DNA, in the replication of DNA, if we talk about replication of DNA in prokaryotes occur in the cytoplasm, but in case of eukaryote, it occurs inside the nucleus. During the replication of DNA, there are many enzymes which are involved, and some of them are, the first enzyme is topoisomerase enzyme. What is the function of topoisomerase? It releases the supercoiling or the tension of DNA, because the DNA in the tensed state is not ready for replication. So the tension has to be released. The second enzyme is important, that is helicase or unwindase. What is the function of helicase? It breaks the hydrogen bonds present between the two strands. That means it unwinds the helix. That is why another name of helicase is unwindase. It is ATP dependent. That means helicase does not do that thing for free, but it needs ATP to break the hydrogen bonds between the two strands. Then the next enzyme is DNA dependent DNA polymerase. DNA dependent DNA polymerase. That means DNA is formed over DNA. That means DNA cannot be formed freely. It has to be, there has to be some template strand on which the DNA will be formed. So DNA polymerase is the enzyme which forms new DNA strands over the existing DNA template. That means the enzyme is DNA dependent DNA polymerase. Let's start. Let's start with the double-stranded DNA. The polarity of one strand is 5' to 3', and the polarity of the other strand is 3' to 5'. And these are hydrogen bonds in between. Okay. First of all, what we need for replication? For replication, we need a very important thing, and that is ORI. ORI. Origin of replication. Origin of replication is a point from where the replication will start. Without ORI, the DNA cannot replicate. Prokaryotes have single ORI, but the eukaryotes have thousands of ORI. Okay. Now, this so ORI is the starting point of replication. It is the starting point of replication. Okay, so now we have a point from where the replication will start, and that is ORI. Now the next step would be the release of the supercoiling. Okay, so these enzymes, they will work simultaneously in the replication. The topoisomerase has released the tension and made the DNA ready for replication. ORI is the site from where the replication will start. Now, let's take the function of the helicase enzyme. The helicase enzyme will come in the picture. Yes. And will start breaking the hydrogen bonds between these two strands. So this helicase enzyme will come and start breaking the bonds. But as we know that helicase enzyme does not do this work for free, it needs ATP for this process. So the cell says, we do not have the enough ATPs to provide to the helicases in one go so that it can open the whole of the length of DNA. Okay, when we make the house, we take loan. We do not have that much of cash to make the whole house at one go. Likewise, to form the new DNA, the cell does not have so many ATPs to provide to the helicase at one go. So, replication fork is formed. So, replication fork is formed. That means that helicase will open some of the DNA and will stop and will say, when you will give me the money, when you will give me the ATP, I'll continue with my work. So some of the DNA is opened up, but some of the DNA is not still opened up. So this is the reason why replication fork is formed, because the cell does not have the enough energy to provide to the helicase in one go. So this is the formation of replication fork. Okay. Then, as soon as the replication fork is formed, we know that the DNA has this habit to form the complementary base pairing. So this replication fork is formed, and to stop the DNA from complementary base pairing again, there are certain proteins which come and bind here, and those are helix destabilizing protein or the single-stranded binding proteins. These single-stranded binding proteins, they prevent the complementary base pairing and they prevent the double-stranded condition to go back again. Okay, so now these two strands will remain single-stranded. This is the helicase enzyme. Now, who will come in the picture? Now, the primase enzyme will come in the picture. Now, let's discuss what primase enzyme does. Now comes the primase enzyme. What primase does? Primase enzyme has RNA polymerase activity. It has RNA polymerase activity. And this enzyme is responsible for formation of RNA primer. This is required for formation of RNA primer. Now, what is this RNA primer? RNA primer is a short stretch of RNA. It's a short stretch of RNA. To understand this, let's take the example of a house. If you want to make a house, a home, then first of all, you'll have to lay down the foundation. Over the foundation, you will make the whole house, right? Likewise, on the DNA replication, on these two strands, we want a foundation. We want a foundation on which the whole strand of DNA can be formed, and the name of that foundation is RNA primer. And RNA primer is formed by the enzyme primase, which has the RNA polymerase activity. Now the question comes in the mind that why we need a RNA polymerase here when we are talking about DNA polymerase? Why not DNA primer can be formed to start the process? Why there is a need of RNA primer? Because DNA polymerase does not have the initiating capacity. Only RNA polymerase can initiate or start the process. So this primase enzyme forms the RNA primer. So the RNA primers are formed here and here. RNA primer gets associated with the DNA strand with its 5' end, and the 3' OH remains free. The 3' OH end of the RNA primer remains free, so that new nucleotide of DNA can come and bind to it. Okay, so the RNA primers are formed and they are added to the opened up DNA strands. What is the function of RNA primer? It acts as a foundation, and the 3' OH end of the RNA primer is free so that new nucleotide of DNA can come and make phosphodiester bond with it. Okay? So now the function of DNA polymerase comes into the picture. Let's see what will DNA polymerase will do now. This is helicase, RNA primer, here and here. The polarity of the upper one is 5' to 3' and 3' to 5'. Okay. Now, the DNA polymerase, what was the function of DNA polymerase? The function of DNA polymerase was to act like a labour. Align brick by brick and apply the cement in between. Likewise, the DNA polymerase will arrange the nucleotides and will form phosphodiester bonds in between. So, new nucleotides are added by and phosphodiester bonds are formed between them by DNA polymerase. Okay, so DNA polymerase attaches these nucleotides and form phosphodiester bond in between. Okay, now let's understand that these nucleotides which are getting added over here, they are charged initially. That means what we can say, that the nucleotides used in the process of replication, they are initially charged. That means DAMP, deoxyadenosine monophosphate, is changed to DATP, deoxyadenosine triphosphate. This is called as the charging of nucleotides. Okay, so these are the charged nucleotides, the triphosphate condition. So NCERT what NCERT says, that these are deoxynucleoside triphosphates. Deoxynucleoside triphosphates, and these are the charged nucleotides. Okay, NCERT says that these deoxynucleoside triphosphates, they serve dual purpose. That means they act as substrate as well as they provide energy for phosphodiester bond formation. That means, understand like this, beta. This nucleotide was a charged nucleotide, okay? That is DATP or DTTP or DCTP, or you can take any one of them. So this is a charged nucleotide, and this is going to be fit here. When this charged nucleotide, gets fit here, then two phosphates are released. Okay, and here, when this would be attached here in the DNA strand, this would be in the form of monophosphate. The nucleotide present in the DNA strand is in the monophosphate condition, but when it came, it was in the triphosphate condition, and when it will be inserted into the strand, then it will change into the monophosphate condition. 2-phosphate would be released, and these will provide energy to DNA polymerase to form the phosphodiester bond. That is why we said that these nucleotides, these charged nucleotides, they serve dual purpose. That means they act as substrate and they provide energy for phosphodiester bond formation. So what is happening over here? These 2-phosphate provide energy. So this process will go on. On the RNA primer, the DNA polymerase will go on attaching new nucleotides and forming the phosphodiester bonds in between them. And during this process, the charged nucleotides act as substrate as well as the source of energy. And this process will go on. But remember that DNA polymerase can do this thing only in one direction, and that is 5' to 3'. And what is that? Let's understand that. Yes, so what we have done up till now? The polarity of this strand was 5' to 3', and of this was 3' to 5'. This was our RNA primer here. This was our RNA primer here. This is the 5' end, and this is the 5' end. The new DNA strand, let's take the yellow color. The new DNA strand is formed here. This is the new DNA strand, and you can see that the new DNA strand will be formed always in 5' to 3' direction. This new DNA strand is formed in 5' to 3' direction, and this new strand will always be formed. That means DNA polymerase synthesize new strand only in 5' to 3' direction. Okay, so these nucleotides will keep on adding, but now we are going to face a problem, and that problem is that this DNA polymerase cannot about turn and go back in the 3' to 5' direction. It can do this work only in 5' to 3' direction. So the DNA polymerase on this strand will stop and say, I will not go back. So the DNA replication on this strand, the strand having polarity 5' to 3', will stop for a while, because we will be needing a new primer. We will have to give a new primer, because DNA polymerase can do this thing only in 5' to 3' direction. And up till now, the cell has taken the loan from the bank, and now it has the money. So it will provide helicase the more money so that it can break the hydrogen bonds. So it will break the hydrogen bonds. And what condition we will have now? Helicase has almost opened up the DNA, and new DNA strand was getting formed but the DNA polymerase is saying I will not go back. So I need a new primer. That means the polarity of this strand is 5' to 3'. The polarity of this parental strand is 3' to 5'. Okay, so this strand will face no problem because it is going in the direction 5' to 3', the same direction as of the DNA polymerase. So the work on this strand will be continued. But on this strand, we will have to provide a new RNA primer. We will have to provide a new RNA primer. So the template strand having the polarity 5' to 3', polarity is to be read in left to right direction. So this template strand having the polarity 5' to 3', on which the strand is getting formed, this strand having the polarity 3' to 5', is not continuously synthesized, but it is discontinuously synthesized. But this strand having the polarity 5' to 3', which is formed on the template strand having the polarity 3' to 5', will be synthesized continuously. So this new strand of DNA is the lagging strand, and this new strand of DNA is the leading strand. Okay, so this one is the leading strand, and this one is the lagging strand. Okay, so the process of replication is not just semi-conservative, but the process of replication is semi-discontinuous also. Replication is semi-discontinuous also. Why it is semi-discontinuous? Because first reason, the two strands of DNA are anti-parallel to each other, and second reason, DNA polymerase can synthesize the new DNA strand only in 5' to 3' direction. Because of these two reasons, the replication process is semi-discontinuous. Okay, so this is the leading strand, this is the lagging strand. The lagging strand, it would be needing many RNA primers. So the lagging strand will be formed in fragments, and those fragments are called as Okazaki fragments. Called as Okazaki fragments. And this Okazaki fragments, they are later on joined with the help of DNA ligase. Later on joined by DNA ligase. Okay. The question regarding the replication, what is the polarity of leading strand, lagging strand, we'll be doing in the MCQ section. Yes. So after the completion of this thing, what will happen? We'll be having this condition. This is the condition. 5' to 3' parental strand, 3' to 5' parental strand, but these two strands, they are the new strands, and you can see that they are not like the parental strands. So this is 5' to 3', 5' to 3', and this is 3' to 5'. Okay, so here there are many RNA primers, but in this strand there is only one RNA primer, but these RNA primer needs to be removed. So now in the end there would be DNA repairing. And what is done during DNA repairing? During DNA repairing, the RNA primers are removed. RNA primers are removed. Proofreading is done. That means if any wrong nucleotide is being inserted in between, that would be taken out and the right one would be inserted again. So proofreading, RNA primers are removed. This is done during DNA repairing, and this is also done by DNA polymerase. And ultimately, after removal of RNA primer, these segments need to be attached again, and this would be done by DNA ligase. DNA ligase. It is also called as the molecular glue, which will form the phosphodiester bond, which will, attach the broken parts after the removal of the RNA primer, and the function of gap filling is done by DNA polymerase itself. Okay, so this is replication. Ultimately, from one DNA molecule, we've got two DNA molecules in which one strand is parental and one strand is new. With the help of the medium, it is formed. So let's within a second, let's again recapitulate the whole process. First of all, we have a double-stranded DNA. We'll start with an ORI, that is origin of replication, from which the DNA replication will start. Topoisomerase will come. It will release the tension from the DNA molecule. Then helicase will come. Helicase will break the hydrogen bonds in between and form a replication fork. After the formation of replication fork, primase, which has the RNA polymerase activity, will form RNA primer, which will act as the foundation and have the free 3' OH end. After that, DNA polymerase will come in the picture, which will add nucleotides in the 5' to 3' direction. And in this process, the charged nucleotides will act as substrates as well as the source for energy. The DNA polymerase will continue doing that, but there will be a problem on the template strand having the polarity 5' to 3', that the DNA can synthesize the new DNA only in the 5' to 3' direction. So one of the strand become lagging because it needs more than one RNA primer, and one of the new strands is leading because it is synthesized continuously. So the process of replication is semi-discontinuous also, because the two strands are anti-parallel and DNA polymerase can replicate only in 5' to 3' direction. Ultimately, this is the condition, and ultimately the RNA primers will be removed. The gaps would be created that would be filled by DNA polymerase itself. The RNA primers would be removed by DNA polymerase itself, and finally that would be glued with the help of DNA ligase. And this is how we will get two DNA molecules. This is replication. Now, after replication, let's go to Transcription. Transcription, again, in case of prokaryotes, occur inside the cytoplasm, but in case of eukaryotes, it occurs inside the nucleus. Transcription means formation of RNA from DNA. Formation of RNA from DNA, any kind of RNA, be it mRNA, rRNA or tRNA. When rRNA and tRNA are formed on DNA, then it is just called a gene. The segment of DNA on which rRNA or tRNA is formed, it is just called a gene. But the segment of DNA on which mRNA is formed is specifically called as cistron. That means you can say that cistron is a segment of DNA, or it is a gene on which it has the information for the formation of polypeptide chain or the protein ultimately. The segment of DNA which will be responsible for the formation of mRNA and ultimately the polypeptide chain has three regions in it, that is promoter region, structural gene and the terminator. Promoter region, structural gene and the terminator. So let's see what is a transcription unit. This is a double-stranded DNA. This is a double-stranded DNA and this is the unit of DNA, the segment of DNA on which the mRNA will be formed, and this is called as cistron or transcription unit. But here, let me make some points clear that transcription is different from replication. How? In replication, both the strands of DNA were acting as template strands. But in case of transcription, only one strand will act as template strand. That means on one of these strands, only RNA will be formed only on the one of these strands, not on the other strand. And second, in replication, the entire length of DNA participates, but during transcription, only a part of DNA participates, which needs to be expressed. So let's discuss this. Cistron, the transcription unit. Here, you can see this is promoter, this is structural gene, and this is the terminator. So there are three parts of the cistron. Here you can see there are two terms. One is the template strand and the another one is the coding strand. Template strand and the coding strand. Coding strand will not participate in transcription, but template strand will participate in the transcription. So coding strand will act as a frame of reference. Frame of reference means if we have to tell that what is the position of promoter and terminator, we will do it with the help of coding strand. So coding strand is the frame of reference. So according to coding strand, the promoter sequences are the upstream sequences. The promoter sequences are the upstream sequences which are present towards the 5' end, and the terminator are the downstream sequences which are present towards the 3' end. Okay, towards the 3' end. So according to coding strand, promoter is at 5' end and terminator is at the 3' end. Now, what is the function of promoter here? Promoter is the RNA polymerase binding site. Promoter is the RNA polymerase binding site. And terminator is the point where the process will stop. And structural gene in between actually codes for the protein. This is the part of the transcription unit which actually codes for protein. Okay, so this is the transcription unit on which the mRNA would be formed. Now, what are the things that would be used during transcription? During transcription, we will be needing RNA polymerase. As in DNA replication, DNA polymerase was required. Here, DNA dependent RNA polymerase will be required. DNA dependent RNA polymerase is required, and this RNA polymerase has the capacity again to take the nucleotides of RNA, align them and form phosphodiester bond in between them. In case of prokaryotes, there is only one type of RNA polymerase. But in case of eukaryotes, there are three types of RNA polymerases. RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 is responsible for the formation of 5.8S. It is responsible for the formation of 5.8S, 18S, and 28S rRNA. 5.8, 18, and 28S rRNA. RNA polymerase 2 is responsible for the formation of hnRNA. What is hnRNA? I'll just tell you. hnRNA is the childhood of mRNA, you can say. And RNA polymerase 3 in case of eukaryotes is responsible for the formation of 5S rRNA, tRNA, and snRNA, that is small nuclear RNA. Okay, so this is the detail of the RNA polymerase that we'll be needing during the process of transcription. So now let's start the process of transcription. This is the double-stranded DNA, and in that whole double-stranded DNA, this is the cistron on which this transcription will occur. Okay, so this is the cistron. First of all, the RNA polymerase will come and bind to the promoter region, because promoter region was the RNA binding site. So RNA polymerase will come and will bind to the promoter region. This RNA polymerase itself here in transcription will itself behave as a helicase. That means helicase is not required during transcription. RNA polymerase will move, will break the bonds, and will form the RNA simultaneously. So this RNA polymerase will move and will break the hydrogen bonds simultaneously and will form the new RNA. This is the RNA formed. But RNA is formed only on the template having the polarity 3' to 5'. Remember this. The polarity of the template strand of DNA on which RNA will be formed is 3' to 5'. And this is the new RNA. This is the new RNA. Okay. And yes, one more point here. The RNA polymerase, during the process of initiation, this RNA polymerase attaches with additional part, that is sigma factor. This sigma factor with RNA polymerase is actually the one who is responsible for the recognition of the promoter sequence and is responsible for the attachment of RNA polymerase over there. When the function of sigma factor is finished, then it is detached. Sigma factor detaches from the RNA polymerase. Sigma factor detaches when the RNA start forming on the DNA. Then the process of elongation, that means the further formation of RNA on this DNA will be continued by RNA polymerase itself. The RNA polymerase itself will continue the process and will form the RNA on this template strand. This is the new RNA molecule. And now when the RNA polymerase is about to reach the terminator site, another factor will come into picture, and that another factor is rho factor. The terminator site is about to be achieved. Terminator site is about to be achieved. So now rho factor will come into picture. And will be attached to the RNA polymerase and will stop the process. The process will be stopped. So, to recapitulate the transcription again, let's start. First of all, RNA polymerase along with the sigma factor will recognize the promoter sequence and will be attached to the promoter sequence. Then the formation of new RNA will be formed on the template strand whose polarity is 3' to 5'. When sigma factor initiates the process, it detaches from the RNA polymerase, and the function of elongation is done by RNA polymerase itself. That is elongation of this new RNA. As soon as the RNA polymerase is about to reach the terminator site, another factor called as rho factor comes and attaches to the RNA polymerase and stops the process. So now the process is terminated. And when the process is terminated, what is the result now? This is the diagram of NCRT. You can see here, sigma factor comes in the picture and it starts the process. The function of elongation is done by RNA polymerase itself, and the rho factor is responsible for the termination of the process. Now, what is the result? What is the conclusion? That we have the DNA molecule on which the transcription was going on, it will again come back to its state. That means hydrogen bonds will be formed again. Hydrogen bonds will be formed again, and DNA will be like what it used to be before. And we have got the new RNA formed over the DNA, and the RNA polymerase will go and will be reused again. Okay, now this new RNA, in case of prokaryotes, is present in cytoplasm, and is called as mRNA. Directly, it is called as mRNA. But in case of eukaryotes, this new RNA is present inside nucleus, and it is not called as mRNA right now, but it is called as hnRNA. hnRNA, or heterogeneous nuclear RNA. Okay, this is also called as primary transcript. This is also called as primary transcript. Okay, so no need of change in case of prokaryotes, because the newly formed RNA is directly called as mRNA. But in case of eukaryotes, this newly formed RNA is called as hnRNA, which is heterogeneous nuclear RNA. This needs to be prepared to transport it to the cytoplasm, because in cytoplasm translation will occur, and the change of this hnRNA to mRNA is called as PTM, that is post-transcriptional modification or the RNA processing. Now let's see what is this RNA processing. Before that, let's discuss what is a monocistron and what is a polycistron. Monocistron, you can say it is a segment of DNA which has the information for formation of only one protein or one polypeptide chain, but the polycistron has the information for more than one proteins. Okay, polycistron is commonly found in case of prokaryotes, and monocistron is commonly found in the case of eukaryotes. Split gene. Split gene means the DNA of eukaryotes has some parts which are expressible and some parts which are non-expressible. The expressible parts are exons and the inexpressible parts are introns. So this gene, the whole gene is interrupted in between with some sequences, and these sequences are called as introns. That means in case of eukaryotes, the introns splits the whole gene, and until and unless these introns are removed from this gene, the translation will not occur. So split gene arrangement is found in case of eukaryotes. Yes, now let's come to the post-transcriptional modifications. They are only needed in case of eukaryotes inside the nucleus, and they are not needed in case of prokaryotes. Now, what is done during the post-transcriptional modifications? Capping. Capping means at the 5' end of the hnRNA, cap molecule will be attached, and this cap molecule is nothing but 5-methyl guanosine triphosphate. 5-methyl guanosine triphosphate. Tailing means addition of 200 to 300 adenylic acid residues. 200 to 300 adenylate residues at the 3' end. So capping is done at the 5' end, tailing is done at the 3' end. Splicing is removal of introns. As I just said, that until and unless introns are removed, translation will not be done. So removal of introns from hnRNA is called as splicing. And there is a term in NCERT which says that the splicing represents the antiquity of the reminiscent of antiquity of RNA world. That means this presence of introns and the splicing, the process of splicing to remove the introns, reminds us that at some point of time, RNA was the king, RNA was dominant, and you have to process the RNA, then only you can form the protein. So you'll have to give attention to the RNA. Now let's discuss what is genetic code. Understand genetic code as a language. Means you want to understand what English is. You want to crack the English language. What you will do? First of all, you will learn the letters of this language, A, B, C, D. And then you will try to make words. And then you will try to learn the meaning of those words. And then you will crack the language. Likewise, there is a language written on DNA, and that language which is written on DNA is the genetic code which we have to crack. And the letters of this language, are only 4. That is A, T, G, C. Only 4 letters are present in this language. And there are total, the words in this language are total 64. And each word contain, and there is a very unique or special point in this language, that each and every word will contain only 3 letters. So each word contains 3 letters. So this language has only 4 letters, A, T, G, C, and the words are total 64, and each word contains 3 letters. And out of them, 61 words has meaning, and the rest 3 words in this language are meaningless. Rest 3 words are meaningless. Now let's go to the scientific language. These A, T, G, C, these are the nucleotides. The letters of this language are called as nucleotides or bases. And the words are called as the codons. And codons is a triplet of, each codon is a triplet of nucleotides or bases. So what we want to have here, we want to understand this language written on DNA. So we'll have to understand the meaning of these 61 words. That means we have to understand that what is that amino acid which is coded by the each codon. Each codon codes for amino acid. That means the meaning of the word is amino acid. And when we'll find out that the 61 codons present in the DNA, for what amino acids they code, will crack the language written on DNA. So what we'll have to do to crack the genetic code? We'll have to know that 61 codons code for what amino acids. So you can say that genetic code is the relationship between the sequence of bases on the DNA and the sequence of amino acids present on the protein or polypeptide chain. So let's discuss what genetic code is. To crack this language written on DNA, or to crack the genetic code, the first work was of Nirenberg. And later on, this work was extended by Hargobind Singh Khurana. Nirenberg, for this work, used only homopolymers. But Hargobind Singh Khurana used homopolymers as well as copolymers or heteropolymers. Copolymers means the repeating unit would not be same. Jaise Nirenberg took only U. That is a homopolymer. So the enzyme which was taken here was Severo-Ochoa enzyme. Very important. Severo-Ochoa enzyme, in another word, is called as polynucleotide phosphorylase enzyme. The speciality of this enzyme is it can form the RNA in vitro conditions. That means outside the living cell without the DNA. It can form the RNA in vitro conditions without DNA. That means this enzyme can work in DNA template independent manner. It is independent of the DNA template. It does not need DNA to form RNA. So what Nirenberg did, he took a solution and provided only UTPs in this solution. Provide only UTPs, that is uridine triphosphate, and along with it, he provided the enzyme Severo-Ochoa. And this Severo-Ochoa synthesized the RNA. the mRNA in this solution without the help of DNA, and this mRNA was poly-U mRNA because the repeating unit was only U, 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 U. And when the translation was done on this mRNA, then it was cracked that U, 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 U is the word, is the codon which is responsible for the formation of amino acid phenyl, which codes for the amino acid phenylalanine. Phenylalanine, which codes for the amino acid phenylalanine. That means we have cracked the first word of this language, that is UUU, and the meaning of this word is phenylalanine. And likewise, using copolymers and homopolymers, Hargobind Singh Khurana extended the work, and all the 64 codons were cracked. Hence, the genetic code was cracked. Now, there's a sheet given over here. There are some important, you can see that there are so many amino acids and so many codons. There are total 64 codons, 61 codons over here, because 64 codons, sorry, are over here, out of which 3 codons are stop codons, which do not code for any amino acid, and 61 are the coding codons. Now, let's move on to the features of genetic code. The features of genetic code. Yeah, the features of genetic code are, first of all, the genetic code is universal. Universal means if UUU codes for phenylalanine in me, then it will also code for phenylalanine in bacteria also, and in all the living organisms. That means one codon will code only for one type of amino acid in all the organisms. Degenerate means that one amino acid is coded by more than one codon, because the words in this language are 64, and out of them the words which has meaning are 61, and the meaning is only the kind of proteins they have to code for, the kind of amino acids they have to code for are only 21. That means in this language there are many synonyms. That means many meanings have the same word, right? So that means one amino acid is coded by more than one codon. That is the degenerate condition. One amino acid is coded by more than one codons. Like arginine, serine, they are coded by 6 codons, right? Then unambiguous or specific. Unambiguous means the genetic code is very specific. That means one codon code only for one amino acid. If UUU codes for phenylalanine, it codes for phenylalanine. It does not code for any other amino acid. So it is very specific. Total codons are 64. Out of them, 61 are coding and 3 are stop codons. AUG, out of these coding codons, AUG is the start codon or the initiation codon, which has dual purpose. NCERT says that this AUG serves the dual purpose. That means it acts as initiation codon, and second, it codes for methionine. It has 2 functions. It codes for methionine, AUG, and also act as the start codon. Start codons means the point on RNA from where the translation starts. Stop codons are UAA, UAG, and UGA. These are the stop codons. 3 stop codons, which are nonsense codons, which do not code for any amino acid. Now let's discuss the process of translation. Translation is formation of protein on mRNA, and be it a prokaryotic cell or a eukaryotic cell, it will be done in the cytoplasm. Now, which cell organelle is responsible for the synthesis of protein? Ribosome. That is why they are called as the protein factory. Okay, now before translation, let's discuss the types of RNA present in a cell. Broadly, there are three types: mRNA, rRNA, and tRNA. mRNA is the template or the messenger RNA which actually codes for the polypeptide chain on which the protein chain is formed. rRNA is ribosomal RNA which has the structural role that it forms the ribosomes. And the transfer RNA is the adapter molecule or the soluble RNA which carries the amino acids during the process of translation. So, ribosomal RNA has structural role. And what is the structural role of rRNA? To form ribosome. And second, rRNA can also act as catalyst or the ribozyme. We will just discuss it now. tRNA carries amino acid during the process of translation. Francis Crick told that there has to be some molecule which understands the language written on DNA on one hand and carries the amino acid on the other hand, and that is the tRNA, which is also called as the adapter molecule. Now, ribosome. Can you tell that what is the size of ribosome in eukaryotes or prokaryotes? In prokaryotes, the size of ribosome is 70S, while in case of eukaryotes, it is 80S. 70S can be broken down to 30S and 50S. 50-30 comes out to be 80, but no, it is 70. And 60-40, 80. 30S subunit is the smaller subunit of ribosome of 70S, and it is constituted of 16 srRNA. 50S subunit is composed of 5 srRNA and 23 srRNA. Then, in case of eukaryotes, the 40S subunit is constituted of 18 srRNA, and the 60S subunit is constituted of 5S, 5.8S, and 28 srRNA. Now, this is the tRNA. You can see this is the clover leaf model of tRNA. There are two models of tRNA: clover leaf model, which is a two-dimensional model, and another one is the inverted L-shaped structure, which is given by Kim and Kluggs. And this clover leaf model is given by Hawley. So what does this explain? What needs to be noted over here, that this tRNA has 4 sides. 1, 2, 3, 4. Remember, this is the anticodon loop. The anticodon loop with which the tRNA will attach itself to the mRNA, and on mRNA codon are present. On mRNA codon are present, the tRNA would be having the anticodon, and this is the site of mRNA binding site on tRNA. At this position, the amino acid will bind to the tRNA. At this position, the enzyme amino acyl synthetase will bind, and at T loop, the ribosome will bind. So that means tRNA is that friend which is responsible for connecting all the friends. That means during the process of translation, all the types of RNA are involved: mRNA, rRNA, tRNA. rRNA is involved in the form of ribosome, mRNA on which the protein is to be formed, and tRNA is the one which carries the amino acid and will attach all of them. At this end, mRNA will be binded. At this end, ribosome will be binded, which contains rRNA, and at this end, amino acid will be binded. So it takes all the constituents together. Now, let's discuss the process of translation. The process of translation is divided into initiation, elongation, and termination. Initiation means the start of the process, and when on mRNA you have to form the protein, then it would be done in 3 steps. First step, the initiation process is divided into 3 steps. In the first step, the smaller subunit of ribosome will bind to the mRNA, then tRNA will come, and then the larger subunit of ribosome will come. Then elongation will be done. I'll be explaining this in detail. Then the elongation comes, and what is involved during elongation? The translocation of this ribosome on the mRNA and transfer of new charged tRNA. And termination is the point where the process will stop. So let's discuss it in detail. First, let's discuss the initiation process. This is the mRNA. On the mRNA, first of all, the smaller subunit of ribosome will bind. Smaller subunit. And you know, in case of prokaryotes, this smaller subunit of ribosome will be 30S, and in case of eukaryotes, it would be 40S. So smaller subunit of ribosome will come and bind here. Then, this is the initiation codon, AUG, start codon. With this start codon, the tRNA will come and bind. The tRNA will come and bind. And you know that this was the anticodon loop, right? So tRNA will bind with the mRNA by the anticodon loop, and this is the codon, start codon present on mRNA. This is the very first amino acid. So this tRNA, the first tRNA is called as the initiator tRNA. The first tRNA which carries the very first amino acid is called as the initiator tRNA. And ultimately, what will happen? The larger subunit of ribosome will come and bind. The larger subunit of ribosome will come and bind. So see, initiation is done. Smaller subunit of ribosome, then the tRNA came carrying the very first amino acid, and then the larger subunit of ribosome. And then the larger subunit of ribosome. Okay? So, what needs to be understood here? That tRNA needs charging before adding to the mRNA. It needs charging. And what is the meaning of charging of tRNA? See, first of all, ATP combines with amino acid. That means amino acids are activated. Remember, beta, amino acids are activated with the combination of ATP. So here, amino acid AMP complex would be formed, and then this amino acid AMP complex will be binding with tRNA. And where? At what point at the tRNA? To the 3' end of the tRNA. So now what we will be getting? We'll be getting a tRNA with which amino acid is associated. With which the amino acid is associated. Okay? tRNA with which amino acid is associated. So this is called as a charged tRNA or it is called as the amino acylation of tRNA. amino acylation of tRNA. So first of all, the amino acids are activated with the addition of ATP, and then that amino acid is added to the tRNA. This is called as the amino acylation of tRNA. That means tRNA which is associated with amino acid. And the very first tRNA is called as the initiator tRNA. So this is the initiation done. Now let's move on to the elongation process. What happens during elongation? There would be translocation of the ribosome over the mRNA. That means this ribosome will move over the RNA. So when there would be translocation of ribosome on mRNA, then new codons will come into picture. New codons will come into picture like UAC will come. Let's take. So new codons will come into picture and we will be needing new charged tRNA. Okay. So first of all, these ribosomes needs to be translocated on the mRNA, and then we will be needing the second tRNA. This is the first tRNA carrying the very first amino acid, and now we will have to take the another tRNA carrying the amino acid second. So transfer of new charged tRNA. So elongation, during elongation what happens? That ribosomes keep on translocating on the mRNA, new codons come into picture, and for those new codons, the new tRNAs come into picture and they are added to this site. I will be discussing this in detail. So transfer of new charged tRNA. Okay, so imagine when this ribosome is getting translocated on this mRNA, this UAC will come into picture and the new tRNA will be trying to get into the ribosomes. This larger subunit of ribosome beta has two sites, two major sites. One is called as the E site, exit site, three major site. One is the P site and this is the A site. So A site is the entry point of the new charged tRNA, and P site is the site where the bond between the two amino acids, that is peptide bond, will be formed. So when this happens, this new charged tRNA will enter here, and what it will say? It will say to the old tRNA that go from here, now I am coming here. So this old tRNA, the old tRNA will take the exit, and now this new tRNA will enter into the P site, and during this process this old tRNA hands over its amino acid to the new tRNA. Okay, so this tRNA is empty now and it will take the exit, and this tRNA has two amino acids now, amino acid 1 and amino acid 2. And in the P site, the peptide bond between the two amino acids would be formed. The peptide bond would be formed. And this is very important, that the energy which would be used in the formation of peptide bond, we have provided this energy in the first phase itself when we were activating the amino acids. Okay, this is important. The energy used in the formation of peptide bond, we had provided this in the first phase itself when we have charged the tRNA and we have activated the amino acid. So and now this is important. Who forms this peptide bond between the two amino acids during translation? The peptide bond is formed by the enzyme peptidyl transferase. Peptidyl transferase. This peptidyl transferase is a ribozyme. It's a ribozyme, and this is very important, very important. This ribozyme is 23 srRNA, very important. 23 srRNA in case of prokaryotes, and this is 28 srRNA in case of eukaryotes. This is very important. 28 srRNA in case of eukaryotes and 23 srRNA in case of prokaryotes. Okay, so this process will go on. The ribosome will keep on translocating on the mRNA. New tRNA will come, the old tRNA will go. The amino acid chain, the protein chain will be growing, and this will continue until the stop codon reaches. So when this reaches the stop codon, so what happens, beta? What happens? This is the last tRNA, because there are no tRNAs for the stop codon. So this is the last amino acid, and this carries a long chain of amino acid, that is the whole protein chain. So when the stop codon comes, there are another factors which come into the picture, and those are called as the release factors. What is the function of release factors? They stop the process when the ribosomes reaches the stop codon. They stops the process by releasing this protein chain and by releasing these ribosomes. So these release factors are also ATP dependent. These are also ATP dependent. And finally, what we get? Finally, we get mRNA, the smaller subunit of ribosome, the larger subunit of ribosome, and we get the protein chain. These smaller and larger subunit of ribosomes, they are used again. Protein chain is used where it has to be used, and mRNA after a point of time degenerates. So this is how proteins are formed. Okay. Yes. UTRs are present on either sides of mRNA, towards the 5' end as well as the 3' end. Before promoter sequence, UTRs are present, and after terminator sequence, UTRs are present. So UTRs are untranslated regions which do not get translated, but they make the process of translation more efficient, and they are present both at 5' and 3' end. Let's talk about the regulation of gene expression now. Regulation of gene expression means, as we know, that ultimately DNA codes mRNA, ultimately mRNA forms protein, which is responsible for the expression. But the expression of gene needs to be regulated. That means whether it has to be stopped or it has to be switched on, there should be regulation of gene expression according to the requirement. In prokaryotes, this regulation of gene expression, that it would be expressing or not, this is very simple in prokaryotes, because in prokaryotes, replication, transcription, translation, everything will be occurring in the same compartment, that is cytoplasm. So the mRNA does not have to go anywhere, and PTMs are also not required in case of prokaryotes. So this regulation of gene expression is very simple in case of prokaryotes, and when a gene needs to be expressed, it has to cross only one hurdle, and that is transcription. If transcription is done, if it is initiated, then no one can stop and the process will go on. That means in case of prokaryotes, the control of rate of transcriptional initiation is the predominant site for control of gene expression. For control of gene expression, that means if transcription is initiated, then the process will go on. It's very simple. But, and one more thing, that transcription and translation are coupled in case of bacteria. That means when the mRNA is getting formed on DNA, simultaneously translation starts, because they are present in the same compartment and RNA does not need any processing. So if transcription starts, that means ultimately translation will happen. So that is why it is simple. But in case of eukaryotes, the regulation of gene expression is a bit complex, and 4 hurdles have to be crossed, and then finally the gene will be expressed. And those 4 levels are first level is the transcriptional level. That means the first hurdle to be crossed is formation of hnRNA. That is primary transcript. Primary transcript is hnRNA. Okay. hnRNA is formed, but hnRNA needs processing, because on hnRNA you cannot go for translation directly. So that means the second step which has to be crossed is the processing of this hnRNA, that is PTM. And the major important step of the PTM is splicing. Then mRNA is formed inside the nucleus, but then the major problem is successful transport of this mRNA from nucleus to the cytoplasm, because sometimes mRNA gets degenerated during this transport. And then the final step which has to be crossed is the successful translation on mRNA in the cytoplasm. So these 4 hurdles has to be crossed in eukaryotes for ultimate regulation of gene expression. Now let's talk about the operon. Operon, the concept of operon was talked about by Jacob and Monod. And this concept of operon is found in prokaryotes. Concept of operon is found in case of prokaryotes. Now, what is operon? Operon is a segment of DNA which is a cluster of genes which contains many genes, a segment of DNA which contains many genes, and it is responsible for some metabolism, like lac operon. Lac operon is responsible for the metabolism of lactose. So the segment of DNA which is responsible for the lactose metabolism is lac operon, and it is a cluster of gene. So let's understand the model of operon. Operon is a cluster of genes constituting of many genes. This first is the regulator gene. The regulator gene or the inhibitor gene. And this is the promoter of I gene. What is the function of this I gene? I gene forms a protein. I gene in this operon is a constitutive gene. Constitutive genes means which will always be open. Regulatory gene always expresses itself and always form a protein, that is a repressor protein. And this is the promoter of I gene, which is responsible for the transcription of the regulator gene. So this regulator gene, what is the function of regulator gene? To form repressor protein. to form repressor protein. Okay, what is this? This is the promoter of structural genes. This is the promoter of structural genes. That means these three which you are seeing here, these are the structural genes, and all these structural genes Z, Y and A, they have a common promoter, that is this. That is why this structural gene forms a polycystron. Polycystron, because all of them are regulated by only a common promoter. And this is the operator site. Operator site is the site of repressor protein binding. Here, the repressor protein formed by the I gene go and binds to the operator site. So, operator site is the repressor protein binding site. It is the repressor protein binding site. Repressor protein binding site. So this is the promoter of I gene. This is the I gene which is always switched on and always form the repressor protein. This is the promoter of the structural genes, which is a common promoter. This is the operator site. Operator site is the site where the repressor protein or the regulator protein comes and bind. Okay, structural genes are the genes which actually codes for the enzymes responsible for the metabolism. Now let's understand the lac operon. Lac operon, E. coli, when present in a medium which has lactose, it will take the lactose and will digest it. Like we take food and then digest it inside our body. Likewise, E. coli will take the lactose from the medium, and when it takes the lactose from the medium, it needs to be digested. That means the operon has to be switched on. So let's first discuss the case when there is no lactose in the medium. When there is no lactose in the medium, the E. coli will not take it, and when the food is not taken, then there is no need of digestion. So, in the absence of lactose, the operon is switched off. And what is the reason of the operon being switched off? Let's see. This is the I gene. The function of I gene is to always form a repressor protein. Okay? This repressor protein, as soon as it is formed, it has active conformation and it has the habit to go and bind to the operator site. So repressor protein will go and bind to the operator site, and the promoter over here will not be able to bind to the RNA polymerase. We know that in order to transcribe these structural genes, the promoter should be binded with the RNA polymerase, right? But because the repressor protein is present here, so there would be loose binding of the RNA polymerase. We know that promoter site is the RNA polymerase binding site. So there would be loose binding of RNA polymerase. And because of this loose bonding of RNA polymerase, this portion will not be transcribed. There will be no transcription and the enzymes will not be formed and the operon will be switched off. The operon is off. So this is the reason why operon is off. Now let's discuss the other case when lactose is present in the medium. And when lactose is present in the medium, it will take the lactose from the medium, and when the food is taken, it needs to be digested also. So the function of I gene is to always form the repressor protein. It will form the repressor protein. But now there is some problem. That means when the lactose is present in the medium, this is the lactose molecule. when it is present in the medium, some of the lactose goes into E. coli. Some of the lactose goes into E. coli cell and binds with this repressor protein. As soon as this lactose binds with the repressor protein, it makes the repressor protein inactive. And this inactive repressor protein is unable to go and bind to the operator site. And when it does not bind to the operator site, that means there is tight binding of RNA polymerase. And when there is tight binding of RNA polymerase with the promoter of structural gene, then structural genes would be transcribed and the enzymes would be formed. 3 enzymes would be formed: beta-galactosidase, permease, and trans-acetylase. These 3 enzymes would be formed, and these 3 enzymes, the beta-galactosidase is responsible for the breakdown of lactose into glucose and galactose. Permease increases the permeability of E. coli towards lactose, and trans-acetylase is responsible for the transfer of acetyl group. So now the operon is switched on. The operon is switched on in the presence of lactose. So that means lactose is the agent over here which is responsible for the opening of the this lac operon. So in the presence of lactose, operon would be on. So what we can deduce from here, that lac operon is an inducible operon. That means it can be induced with the presence of an inducer, and that inducer is lactose or allolactose. Okay? And this works under negative regulation. What does that mean? This works under negative regulation. That this means that the regulator protein or the repressor protein formed by the I gene always has the intention to switch off the operon. This is the negative regulation. And this operon supports the catabolic pathway. That means the lactose is broken down when the operon is on. And yes, one more thing, that a low amount of expression of operon is always there. That means I've just told you that in the absence of lactose, the operon would be switched off. Yes, it is switched off, but a little amount of expression is present even in the absence of lactose, because a little amount of permease has to be present all the time in the cell, because if permease would not be there, how the lactose will come inside the E. coli and the operon will be on? So a little amount of permease will always be formed, and a little low amount of expression will always be present even in the absence of lactose. Okay, let's discuss the Human Genome Project. Human Genome Project is one of the biggest projects in the history of biology, and it is a 13-year mega project. It started in the year 1990 and was ended in the year 2003. It was coordinated by US Department of Energy and National Institute of Health. So officially, or you can say that the project was the deadline of the project was 2003, but actually the chromosome 1 is the largest chromosome. So it took some time for the sequencing of chromosome 1, and the sequencing of chromosome 1 was completed in May 2006. May 2006. Okay. So what are the goals of Human Genome Project? The goals of Human Genome Project were to identify all the genes which are present in human DNA, to determine the sequence of the 3 billion base pairs present in the human DNA, and to store this information in the form of databases, and to improve the tools for data analysis. Whatever the sequence present on DNA, we should have to analyze that also. And it has to also deal with the ELSI, that is ethical, legal, and social issues. Now, what were the major approaches which were taken during this Human Genome Project? There were 2 approaches. First was the ESTs, and the second approach was the sequence annotation. Now, what are they? That means ESTs is the technique where only those parts needs to be sequenced which are coding parts. That means which are expressed as RNA. That means one methodology was like, we will only sequence those parts of DNA which are coding and which are expressed as the RNA. But sequence annotation says that we will code the whole DNA, and then later on we'll assign that these are the coding parts and these are the non-coding parts. So obviously this is better. Let's sequence the whole DNA, and then later on we will annotate that these are the coding regions and these are the non-coding regions. Okay, so what was done for the Human Genome Project? First, DNA was isolated, and then after the isolation of DNA, it was broken down in the fragments. Because it is very difficult to deal such a long length of DNA in one time. So it was broken down into fragments, and each fragment was amplified to form the several copies using the host, that is E. coli or yeast. And the vectors used for the amplification of each fragment of DNA were bacterial artificial chromosomes and yeast artificial chromosomes, that is BACs and YACs. And later on, these fragments were sequenced with the help of automated DNA sequencer, and this automated DNA sequencers, they were based on the technique developed by Frederick Sanger. After the revelation of the findings of the Human Genome Project, what was found that the human genome contains 3164.7 million base pairs, and it says that the average gene, it says that a single gene generally constitutes of 3000 bases. A single gene generally constitutes 3000 bases, but there are some exceptions also, like the dystrophin gene. Thus, dystrophin gene is the largest gene containing 2.4 million bases. That means this gene, a single gene contains 2.4 million bases. Otherwise, generally, our gene contains 3000 bases. The total number of genes found in the DNA was estimated at 30,000 or much lower. But previously, before the Human Genome Project, it was thought that the total number of genes found in DNA can be 80,000 to 1,40,000. But now it is known that no, they are estimated at about 30,000. And yes, one more important thing, that we all are similar. In 99.9%, we are all similar. There is dissimilarity in only 0.1% part of DNA. Then it revealed that we have just know the function of only the 50% of the genes which are discovered. Matlab, the function of the genes is still, some of the genes are still need to be, the function of those genes still need to be discovered. Yes, it says that more than 98% of DNA is junk DNA, which is not the coding part, and less than 2% of the genome codes for protein. Okay, and repeated sequences make up the large portion of the human genome. That means human genome contains several portions which have repetitive sequences on them. Okay. And these repetitive sequences, they are not coding, they are the non-coding parts, but they have a very important role to play in the process of evolution because they decide the process of polymorphism. Yes, the chromosome number 1 is the largest chromosome, contains 2968 genes. Learn with me, 2968 gene. And the chromosome Y is the smallest chromosome. It contains only 231 genes. Then there is one more thing, that is single nucleotide polymorphism. What is single nucleotide polymorphism? That there are around 1.4 million locations on DNA in which we all differ from each other only in one nucleotide. And there are how many locations on DNA? Such locations, 1.4 million locations. That is called as SNPs or SNPs, single nucleotide polymorphism. Now let's come to the last part of the chapter, and that is DNA fingerprinting. As we know that 99.9% similarity is there in between us humans, and there is only, the difference is only 0.1%. Okay, we all differ from each other only in 0.4% of the base sequences, and that comes out to be, what is the 0.1% of 3 into 10 raised to the power 9? That comes out to be 3 into 10 raised to the power 6. See, 0.1% of 3 into 10 raised to the power 9 is 3 into 10 raised to the power 6, and this is also very large. So what we did with this DNA, 3 into 10 raised to the power 6 base pair, we did density based centrifugation, that is with the help of cesium chloride. And what we found after doing the centrifugation, we found major part of DNA, that is the bulk genomic DNA, and a minor peak of DNA, that is the satellite DNA. That means in this 3 into 10 raised to the power 6 base pairs, some of the portions are the portions which forms the repetitive DNA, and that DNA is called as the satellite DNA. The satellite DNA is the repetitive DNA found in the human genome. So satellite DNA is the repetitive DNA. This repetitive DNA means a particular sequence repeat itself many a times in this particular portion of DNA, that is repetitive DNA. And this repetitive DNA can be a mini satellite or it can be a microsatellite. If the repeating units are around 1 to 6 base pair long, that means around suppose 4 base pair repeat themselves many a times, that would be a microsatellite. If it is greater than that, if it is more than, the repeating unit contains more than 10 base pairs, then it is a microsatellite. Okay, mini satellite VNTRs, variable number of tandem repeats. The example of mini satellite is VNTR, and the example of microsatellite can be SSRs and STRs. STR stands for short tandem repeats. And SSR stands for short sequence repeats. Or you can say single sequence repeats. Okay, so in the microsatellites, the repeating unit is smaller, and in the mini satellite, the repeating unit is a bit larger. So let's start with VNTR, variable number of tandem repeats. What does it mean? This forms the basis of DNA fingerprinting. This forms the basis of DNA fingerprinting. What is that? Let's understand this. Suppose there is a sequence A, T, G, C, T, T, A, T. This sequence C, T, T, A. Suppose this sequence repeat itself many a times on a part of DNA. So this part of DNA will be repetitive DNA, right? Suppose this particular sequence is repeating itself for 4 times. So 4 times. So this part would be the repetitive DNA. Okay. This sequence is the repeating sequence of VNTR. And this repeating sequence is same in all of us, but the number of repeat of this sequence differs in all of us. That means, suppose for instance, on my chromosome number 1, this particular sequence repeat itself for 4 times. And suppose in another person's DNA, it repeats itself for 5 times. That means the sequence is same, but the number of repeats of this particular sequence are different in all of us. That means no two person can have same VNTR. Everybody would be having different or variable number of repeats on their DNA of the same sequence. This forms the basis of DNA fingerprinting. Except the monozygotic twins. No one can separate monozygotic twins because even DNA fingerprinting fails over there. So DNA fingerprinting, what's the basis of DNA fingerprinting? VNTR. No two person except monozygotic twins can have same VNTR. Everybody has different number of repeats. Now let's discuss the process of DNA fingerprinting. First of all, we'll have to isolate the DNA. So first of all, we'll have to do isolation of DNA. Suppose we have reached some crime spot. Okay, suppose Daya has reached the crime spot and he has taken a hair follicle from there. And now we have to know that who is the criminal. So first of all, we'll isolate the DNA from that hair follicle. So first step is the isolation of DNA with the help of several enzymes. Then second, treatment with restriction endonucleases. Treatment with restriction endonucleases. Now, just understand one point, that this repetitive DNA, the repetitive DNA is flanked with or is bordered with certain restriction sites. Certain restriction sites. Okay, so this repetitive DNA would be having one restriction site over here and one restriction site over here. And these restriction sites are identified by the restriction endonucleases. They will come and cut the DNA from here. That means when these restriction endonucleases will come and make a cut, then you will have many fragments. And some of the fragments would be having the repetitive DNA and some of the fragments would not be having the repetitive DNA. Suppose this part is not repetitive. So when it will make cut, this part will be having the VNTR, this part will be having the repetitive DNA, but this part will not be having the repetitive DNA. So what is the result? What is the result of treatment with endonucleases? We would be having many DNA fragments. We would be having many DNA fragments. Some of the DNA fragments have this VNTR and some of them do not have this VNTR. Okay, and this technique is called as RFLP. RFLP, that is restriction fragment length polymorphism. Restriction fragment length polymorphism. That means I just told you that no two person can have same VNTR. The length of the VNTR, the length of the repetitive DNA would be different in every organism. So when this restriction endonucleases make a cut, the fragments, the length of the fragments of DNA would be different in each individual, and that is restriction fragment length polymorphism. Okay. Now the next step would be the separation of fragments. in the test tube, we would be having various fragments now. Those fragments needs to be separated. So now separation of fragments, and this separation of fragments is done with the help of a technique called as gel electrophoresis. So with the help of gel electrophoresis, the fragments of DNA are separated on the basis of their molecular weight. Then next, what happens that we have many fragments of DNA on the agarose gel. Then there would be a denaturation process. What will happen during the denaturation process? We will dip these fragments in an alkaline solution. And in the alkaline solution, what will happen? The double-stranded fragments of DNA will be converted into single-stranded fragments of DNA. So now we have many single-stranded fragments of DNA. Then what is done? After that, southern blotting is done. What is southern blotting? Southern blotting is transfer of these DNA fragments on a nitrocellulose membrane or nylon membrane, nitrocellulose membrane or nylon membrane for easy handling. So after separation of DNA fragments, there is denaturation process. We have converted all the double-stranded fragments into single-stranded fragments, and then southern blotting. After southern blotting, what we will do, this step is important. Hybridization. And to understand hybridization, first understand what is a DNA probe. Which color I should take? Let's take this one. What is a DNA probe? A DNA probe is an artificially synthesized DNA. We synthesize this DNA in vitro conditions. It is a single-stranded DNA, and the sequence of this DNA is complementary to the sequence of the VNTR sequence. And the most important thing is that this DNA probe is radioactively labeled. And you know why we do radioactive labeling? Fluorescence, frescence. So the DNA probe is artificially synthesized, radioactively labeled, single-stranded DNA which has complementary sequence to VNTR sequence. Okay. Now imagine that on that nylon paper or the nitrocellulose membrane, we have many single-stranded fragments of DNA, right? Many single-stranded fragments of DNA. And I just told you that in some of the fragments, there is repetitive sequence, and in some of the fragments, there will be no repetitive sequence. So when we will take this DNA probe and will provide it here, then hybridization will be present where there is the repetitive sequence. Matlab, those fragments those fragments which do not have the repetitive sequence or the VNTR sequence, they will not show hybridization or attachment with the DNA probe, but those parts which have the repetitive sequence, they will show the attachment, they will hybridize, they will form complementary base pairing with the DNA probe. So those parts which have the repetitive sequence now are attached with a radioactively labeled substance. And then finally, so this is repetitive DNA hybridized with a DNA probe. This is repetitive fragment of DNA hybridized with DNA probe. And these are the fragments which do not contain repetitive sequence. Now the last step is autoradiography. What was the number of the previous step? It was 6. Okay. So the last step is autoradiography. Auto means automatically, by itself. Those fragments of DNA which are hybridized with their DNA probe, and DNA probe is radioactively labeled. So when this is exposed with the X-ray, the fragments of DNA which were hybridized with the DNA probe, they will show fluorescence and they will show I'm present here. So we'll get a certain pattern. So autoradiography is that the hybridized fragments will show fluorescence by themselves. Will show fluorescence by themselves when exposed with X-rays. Okay. So this is autoradiography. So we'll get a particular pattern after audio radiography. Suppose we are getting these bands, like these bands. So these bands are unique for every individual except the monozygotic twins. So this is how we use this DNA fingerprinting to know the real identity of a person, and that is DNA fingerprinting. There is a diagram of NCRT given over here. You can see that we have taken this chromosome, we have taken this DNA from the crime scene, and we have taken the DNA of 2 suspects, that is suspect A and suspect B, and we have done the whole process with the genome of A and the genome of B, and we have got these bands. We have got these bands. 2 bands are these, 2 bands are these, 2 bands are... So now we can compare that the DNA taken from the crime scene shows the same pattern as that of the individual B. So now Daya has got the person and B is the criminal. So this is how DNA fingerprinting is used to resolve the this forensic issues and to resolve the paternity issues also. So now the chapter is finished, and I hope that it would help you in every possible manner to understand the topic, and it will help you all to give your term 1 exam. And thank you very much for the patience, and all the best for the term 1 exam. Thank you.